Hi all, welcome to lecture 10 on bacterial classification. Okay, let's look at some definitions. So first is the taxonomy. So taxonomy is a science of biological classification and usually is provide an ID or name and place for them in a group with similar characteristic we call as a taxa. Okay, taxonomy is useful for classification, nomenclature, and identification. Classification is an arrangement into groups based on mutual similarity or revolutionary relatedness. Nomenclature is a branch of taxonomy concerned with assigning names to taxonomic groups. So literally mean you are giving names to a particular organism. Identification is an application side of taxonomy, which is a process of determining specific identity of an isolate. Okay, binomial nomenclature is a scientific name which is a Latinized form. So all the scientific names should be written in Latinized form, and that's we call as a binomial nomenclature. So the science of taxonomy, which is based on binomial nomenclature, as I mentioned earlier. And then this is invented by Carolus Linnaeus in 1757, and then he is also known as a father of taxonomy. So in scientific name should be written in two. So usually contain two names, which is genus name and species name. So both names should be italic. And then the first letter of genus name should be capitalized. So for instance, Staphylococcus aureus, the S should be capitalized. And also the first genus name can be abbreviated. For example, Staph for Staphylococcus aureus, or can be S. aureus. And the second name is the species. The species name should be lowercase and never be capitalized. For example, aureus. And then this second name also shouldn't be abbreviated. And then when you're writing in a document, it should be underlined as it's shown here. And if it's a printed document or typed, it should be italic as it's shown here. Okay, let's move on to the classification system of organisms. Okay, in 1935, Carolus Linnaeus, the father of taxonomy, so he came up with a two-kingdom system, which is including Animalia and Plantae. Okay, however, in 1866, a German biologist Ernst Haeckel came up with a three-kingdom system, which consisting of Protista, which is all unicellular organism, which including bacteria and other protozoa, and then you also have Plantae and Animalia kingdom. Okay, in 1969, R.H. Whittaker came up with a five-kingdom system, which is including Animalia, Plantae, fungi, Protista, and Prokaryote. And then you have to take note that viruses are not included in this five classification kingdom because viruses are considered as a not living thing. And sometimes that's why people calling them as a virus particles. Okay, biotechnological advances has led to the proposal of three-domain system, and this system is proposed by Carl R. Woese in 1978. And this system is based on the phylogenetic classification, which is works based on the comparison sequence of rRNA and then the cell wall structure. So in the three-domain system, so we have eukarya. So this eukarya will contain all the eukaryotic organism, which under the kingdom Animalia, Plantae, fungi, and Protista. And the second domain is the bacteria, which is a unicellular and cell wall contains peptidoglycan. For example, eubacteria and cyanobacteria. And the last domain is the archaea. So archaea is an ancient bacteria which live in extreme environments. They are also unicellular and no peptidoglycan. And examples of archaea are methanogens, extreme halophiles, and extreme thermophiles. And you also should take note that from evolutionary point of view, archaea is more related to eukarya than the bacteria. Okay, this is a summary. So as we know, usually cells can be eukaryotes or prokaryotes. And then these prokaryotes we have archaea domain, bacteria domain. And then we also have eukarya domain, which is consisting of plants kingdom, fungi kingdom, animals kingdom, and protist kingdom. And then the monera kingdom will falls under archaea and bacteria domain. Okay, let's look at the taxonomy hierarchy. So I believe you guys learned this in your high school or matriculation. So usually a hierarchy starts with a domain and then followed by kingdom, followed by phylum, or in plural we call phyla, and then followed by class, order, family, genus or genera for plural, species or species in plural. Okay, this is an example. So for instance, so the kingdom is bacteria, the phylum is Actinobacteria, class is Actinobacteria, and order is Actinomycetalis, family Mycobacteriaceae, and genus become Mycobacterium, and the species is tuberculosis, and which give to the name Mycobacterium tuberculosis, or you can also write as M. tuberculosis. Okay, let's look at the basics of bacterial classification. So the bacterial classification can be done either phylogenetic or phonetic. So if it's a phylogenetic classification system, means it's a group, reflect genetic similarity and evolutionary relatedness. If it's a phonetic classification, it is merely based on the observable characteristic. It's nothing doing with the genetic similarity or evolutionary relatedness. Okay, a little more definition on the genus and species. So if it's a genus, means it's a closely related species. Species is a collection of microbial strain that shares many properties and differ from other group of strains. Okay, bacteria also can be further divided by strain and type. So strain means it's an organism within a species with varying in a given quality. If it's a type, means an organism within a species with varying immunogenicity. So in conclusion, if it's a species, it is a basic unit of taxonomy, which is representing a specific recognized type of organism. If it's a strain, it is a genetic variation within a species. If it's a type, it is based on the immunogenic variation within a species. Okay, let's look at the various methods of bacterial classification. Okay, the first method of bacterial classification is via morphology. So the morphology can be done either microscopic or macroscopic. So microscopic usually measured as a size, shape and arrangement. For example, either is coccus or bacillus. Either they can form cluster or disperse. And on the other hand, the macroscopic such as colony appearance, example shape, margin and elevation. Okay, this diagram shows a description of colonies on agar plate. So you can study a bacterial colony based as a whole colony such as circular, rhizode, irregular and filamentous as shown here. Or you can study the edge or the margin of the colony such as entire, undulate and lobate, which has been shown here. And sometimes people also studying on the elevation of the colony, whether it flat, raised, convex and umbonate, which is shown here. And sometimes you can also study on the surface, whether it looks smooth and glistening or rough, wrinkled and dry or powdery. Okay, the second way of bacterial classification is chemical or biochemical characteristic. So usually chemical characteristics usually performed by using reagents or staining kits. So for example, a cell wall composition can be studied using a gram stain, and people also using nucleic acid kits to study the nucleic acid composition. And sometimes people also study the storage inclusion, which we studied in our second lectures, and people also doing the pigmentation can be performed by the bacteria. Okay, the third way of bacterial classification is nutritional pattern. So you can identify or classify bacteria based on whether they are autotrophic, heterotrophic or fermentative. So you can identify based on their energy source and also the carbon source. So the fourth way of bacterial classification involving physiology. So this is, for instance, based on the optimal temperature and pH for bacteria to grow. It can also be done via oxygen requirement, whether they need oxygen or not. Also can be done whether they need salt or not. Or is there any specification on the osmotic tolerance? And apart from that, they also can be done via the sensitivity to a particular antibiotic. So apart from that, the classification also can be done based on the serology property. So the serology property based on the antigenic property of a particular strain, and mostly is done to determine a particular strain, serotype and serogroup of some species. The method is based on genetic and molecular analysis. So this is solely based on biotechnology. So the first way you can done based on the G and C composition. So in other way, you scatter all the DNA from the bacteria and then you do the sequencing, and then you measure the total DNA content. So for example, you can do the G and C ratio to A and T. So based on this, you can find the relatedness between two genera or families. And the second method is DNA hybridization. So this method is based on the ability of a DNA strand from one organism to hybridize with another organism. So it's more hybridized it is, so the more related the two organisms are. And the following method is sequences of bases in rRNA. So this way you extract all the rRNA from the organism, and then you sequence them, and then you study how similar or related the sequence to the another living organism. If it's more similar, they are more related. Okay, the following technique is phage typing. So the phage typing involving bacteriophages, which are bacterial viruses. So they are very specific in choosing their bacterial host. So therefore, based on their choosing nature, so we use them to study the susceptibility of certain bacteria to a specific group of bacteriophages to determine their strain. Okay, what people usually do is that they make a thin layer of bacteria on the agar, and after that they put the known bacteriophages on the top of the agar. So as I mentioned before, bacteriophages are very specific, right? So if the bacteriophage can infect the bacteria that put it on the agar, you will get a clear zones. So if it doesn't, you won't get a clear zones. Okay, we can also do a bacterial classification based on their protein content, which is known as a protein profiling. So what do we usually do? You extract all the proteins from the bacteria, and then you run a electrophoresis, and then you'll get a bands like this. So based on the bands, you can classify the bacteria according to the profiles, or usually these profiles are known. Okay, thank you, and please post your questions in the e-learning.